Okay, we're going to do a little run through of the kidney, structures in the kidney and the reason for it. So the first question is from here. Let's just get myself a nice blue pen. Why do we pee? Why do we urinate? So what could be reasons that we urinate? And the first thing is that it's about water. So it's about the regulation of water levels. And the other thing that it's about is removal of waste products. And because we are removing waste products, and these have to be metabolic waste products in order to be excretion. So if we have things that are egested, egestion is not the same as excretion. So our kidneys are involved in excretion. Egestion is just the removal of undigested. Whereas here, if we've got the removal of waste products, then that is excretion. So there's our definition of excretion. It's the removal from the body of waste products of metabolic pathways. And so we can see a little bit in the picture beneath about what the urine is removing. And it is the breakdown products of either nucleic acids, so in that case we're talking about DNA or RNA, and that can lead to amine groups because they have nitrogenous bases. And then the other thing is proteins, because amino acids, nacho cheese, they have an amine group, and that amine group needs to be removed as well. Just a quick recap of amino acid structures. So proteins are broken down, and then we need to remove the amine group. Because we're removing the amine group, this is deamination. Okay, so deamination takes place in the liver. We get deamination in the liver, and that's the removal of the amine group. So if we have a look at the picture here, we can see that there are different ways of doing it. If you're a bird, then you don't pee. So you don't get peed on by birds, you get pooed on. And the reason is they make uric acid, and this forms crystals. And so birds kind of excrete a paste, a uric acid paste. If you're aquatic, if you're living surrounded by water, then you can excrete ammonia. You don't have to combine it. You can see here that in urea, it's being combined with CO2. Not a full CO2, but we can take CO2 and we can take amine groups and combine them into urea. So it's now a waste product of both respiration in the CO2 and a waste product of proteins for excess proteins. Now we're going to have a lot of excess proteins in the body because proteins have a lifespan and cells have a lifespan, and then they need to be broken down through deamination by the liver and then excreted. So the way that we do it is we combine it into urea. And we do that because ammonia is toxic. This is too toxic to be excreting on land. If you're surrounded by water, then that's fine. But if you're not, then it's better to excrete it as the less harmful, less toxic urea. Okay, we're going to talk about osmolarity here. And so here's a definition for us. Osmolarity is the total concentration of solutes dissolved in a solution. So if we think about solutes in a solution, then we could be talking about lots of things which are dissolved in that solution. So what things could they be? They could be things like salts, and the other major one is sugars. So we've looked at salts and sugars. They are going to influence the osmolarity. So we saw osmolarity when we looked at potato osmolarity, the total concentration of solutes. So over here in that box, that would be a high solute concentration, whereas outside there could be a low solute concentration. And actually in the case of seawater is often very salty. So it could be that the concentration in another organism was less than that. Or it could be that the concentration could be the same. So there are two different major types of organisms. One type of organism, and let's see if you can work out which one is which here. One type of organism maintains a constant internal osmolarity, and another, or other types of organisms, they just take the concentration that's around them. So see if you can work out from these two, let's call them A and B, which one takes the concentration of the solution surrounding it and just has the same, and we call them an osmoconformer. So osmoconformer, that means it's the same concentration as the solution it's in. And one of them is going to be an osmoregulator. That means it's going to keep its own concentration of solutes, its own osmolarity. And think about as well which one we are. Are we an osmoregulator or an osmoconformer? Okay, well the answer is that we are osmoregulators. We regulate our solute concentration. It doesn't matter what kind of liquid environment we're in, we still have the same concentration. And it was a bit of a trick question because up here as well, they are both osmoregulating. They aren't, we can see that there is exchange of ions in both. There's an excretion of either dilute or concentrated urine. Both of these are actually regulating their environment. If it was an osmoconformer like a starfish, we wouldn't see these pumping, we wouldn't see ion movement. We would just kind of see, we would see just movement, passive movement, moving in and out, not the creation of urine, which might be different from the environment. So we can see this one here, we can see it's drinking seawater and then it's excreting, it's actively pumping out ions to regulate its environment and making a salty concentrated urine. Now this one over here, although you couldn't actually see it, this says this should be a fresh water. This is a fish in fresh water. So in fresh water it excretes a dilute urine because it doesn't want to lose all of its ions to fresh water, which doesn't have much salt in it. Okay, so here's just a quick recap then. So we talked about an osmoregulator. That is an organism that maintains a constant internal solute concentration even when it's in the environment like sea or if it's in fresh water, and that would be us, all marine animals, bony fish. If you're an osmoconformer, then you have the same concentration, the same osmolarity as the environment, and that would be e.g. jellyfish and some sharks as well. Okay, one of the things you have to be able to draw is a labeled diagram of the kidney. So you do it in a kind of bean shape, and then you need to think about having one layer for the outside, a second layer inside, and finally a third which pulls together. So we could label these as 1, 2, 3. And I would pause the video now to draw the general outline. You need to draw yourself the 2 blood vessels which are connecting as well. And then let's go ahead and label it. So the outermost part of the kidney, like the outermost part of the brain, is called the cortex, the kidney cortex. And just like in the brain as well, we can think about the middle part being the medulla. And after that, we then get into the part which is called the pelvis. And this is where the urine is actually formed. So we go down here, and the pelvis will lead to what's going to connect to the bladder, which is a ureter. So the ureter, this is going to go off to the bladder. And then it will go out through the urethra, which is where urine actually exits the body. Okay, so that's the diagram that you need to be able to label. And we want to also think about next what different things are happening in the different parts of the kidney. So let's have a look at the question which is just at the bottom now. So what's the role of the kidney in mammals, including humans? Well, we've got 2 blood vessels, and actually we should have labelled those as well. There are 2 blood vessels which connect. Because it's to do with the kidney, it's renal. So the renal artery, and over here we have the renal vein. So blood's flowing in, it's going through all the structure of the kidney, and then it's coming out. So we want to think about, well, what does it do then? What's the difference? Well, we said that it's creating, what's coming out here is excretion products. It's nitrogenous waste. We want to get rid of nitrogen-containing waste in the form of urea. And the other thing that we want to get rid of as well is excess water and excess salts. And finally, the other role is the removal of toxins. So we're going to remove toxins and they're going to come out in the urine. And that means that we get an increased level of toxins in urine compared to blood. So it can be concentrated in the urine. Okay, so if it's removed those things and if it's an active working tissue, we can think about what would be the comparison between the blood going in versus the blood going out. What things would they differ in concentration? So just pause there and have a think and see if you can write those down. So going into the kidney, we're going to have oxygenated blood. So going in, there's going to be a higher level of oxygen. So higher levels of oxygen going in, and versus going out, well, so we're going to say increased O2. Going out, there's going to be a decreased O2 level. Why? Because it's used in respiration. So the kidney is a rapidly respiring tissue. It's using a lot of energy for active transport, and we're going to see that in what it's doing with ions and salts and stuff. Okay, so it's doing active transport. What else is going to be in higher levels? Well, usually going in versus out, there's going to be a higher level of water because we're going to remove some water always. There would also be a higher level of urea because that's what it's supposed to be getting rid of, and there would be a higher level of toxins and a higher level of salts. So basically all these things down here that it's going to remove, they're going to be in higher levels, and over here they're going to be in lower levels. And so by doing this, the kidney is achieving osmoregulation. It is regulating water content. And because it's removing water, it's going to regulate the solute content, the osmolarity. So this is just a quick recap then. So the role of the kidney was in the homeostasis of blood sugar, salt and water concentration, called osmoregulation. It filters and removes nitrogenous waste products from the blood, including urea. It also removes excess salts and excess water, and it produces urine to excrete nitrogenous waste. The concentration of the blood coming in is going to have higher oxygen, urea, salt, water and toxin concentrations because they're removed, and those would all be lower. The oxygen is removed for use in respiration, so the blood leaving it in the renal vein is going to be deoxygenated. But some things are going to remain the same, and that's plasma proteins. And that's because plasma proteins are too big to fit through the filter. So we're going to come on to that. The kidney is working as a filter, and things like blood plasma proteins are not filtered. Blood glucose concentration is kind of similar, but it does decrease. So there's not a huge change in blood glucose, but some glucose is being used by the kidney. So the first part of what the kidney does happens up here in the cortex. So blood flows in, and it makes its way all the way up to the cortex, and that's through, we said it came in through the renal artery, but then it will be splitting into smaller arterioles. So they're small arteries. It's going to split into small arterioles, and then it's going to go through the first step. And the first step is ultrafiltration. So the ultrafiltration that we're going to look at is taking place in the outer part, in the cortex. The next part that we also get happening in the cortex is selective reabsorption. So part 2 is reabsorption, taking back things that these are needed by the body. We might need the salt, we might need the water, we might need the glucose. In step 3, we're going down into the medulla, and this is where osmoregulation happens. This is basically the absorption of water, or further absorption of water. And then finally down in here in the pelvis, the white bit, that is where the real excretion takes place. That's where the urine is, and then that's going to pull together. So the overall functional unit of the kidney is called the nephron, and so there are millions of these in each, or around about a million in each kidney. So we've got millions in both kidneys combined, and you can see that they're quite long because they're stretching all the way up from the cortex, this region at the top here. They're stretching all the way up from the cortex, and it's going all the way down into the medulla. So the whole nephron, if we just follow the path, the nephron includes this part. It does a wiggle, it then goes down, back up, does a wiggle, and then down again. So it's a very kind of convoluted path. And this is to give it plenty of time to do some of these active processes. So let's go through and label some of these then. So the first thing that we're seeing there is kind of like, it looks a bit like a cup structure, like an old school cup. And so because of that, it's sometimes called a cup, Bowman's cup, or capsule. So this first bit here, the whole bit, A, that is Bowman's, or the renal, to do with the kidney, capsule. So this is where we're going to get the first process, which is ultrafiltration. And ultrafiltration, if we think about a filter, this is that process where we're going to have gaps and we're going to get big things stuck because they can't get through, but we're going to have smaller molecules which can pass through. They will pass through those gaps. So ultrafiltration, and that's happening. That's the first bit that's going to happen in Bowman's capsule. So the blood flows in, and then it gets filtered, and it flows out. And you can see that then the blood, it takes a path. I'm just following the blood now. The blood now goes round and gets close to the rest of the nephron. And that's so that we can have things being absorbed from this tubule into the blood, from this tubule into the blood, from this tubule into the blood, or that tubule into the blood. So the whole purpose after the filtration is to get things back into the blood, and that's why we've got these rich capillary networks to absorb things back into the blood. So the next thing that we can see there, Bowman's capsule, we can now see this B. We can see a knot of blood vessels. So this knot of blood vessels is the glomerulus. The glomerulus is a knot of blood vessels in Bowman's capsule. Okay, up to C, we can see that this is near. It's quite near to Bowman's capsule. And so the word in biology for near, for C, if you are nearby, then you are proximal. That means nearby. And the tubule goes backwards and forwards. It doesn't take a straight path. So the word for this is convoluted. So it's the proximal convoluted tubule. Proximal convoluted tubule, the PCT. Okay, then we go down to D. Now this, because it goes down and back up again, it's going all the way down, all the way back up again in a loop. This is the loop of Henle. So the loop of Henle goes all the way down and all the way back up. And this is where we're going to get a lot of absorption of the remaining bits of water. So this is where we said in the previous bit, we're now down into the medulla. This is where osmoregulation is taking place. Okay, up to E, and we don't really talk about what it does that much, but because it's further away, it's far, it's distal. So distal, and it's the same. It's a distal convoluted tubule, so, or the DCT. Finally, for F, we have the collecting duct. Because what's it collecting? It's collecting urine. And this, the collecting duct, is where we get our hormone ADH acting. ADH is a hormone that's going to act on this collecting duct. And this can either absorb more or less water. And this is what creates concentrated urine or dilute urine. Okay, so here's our completed picture with all the labels of it on there. And we've got a number of different processes then. So we talked about number one is ultrafiltration. Number two, we're going to take things back that we want. So we reabsorb some things, selective reabsorption. Things like glucose, things like salt need to be reabsorbed. Things like water need to be reabsorbed because we don't want to just excrete all the glucose. In fact, if you do have glucose in your getting into your urine, it means you are diabetic. It means there's so much glucose that it can't be selectively reabsorbed in the proximal convoluted tubule, and it makes its way into urine. Okay, down into number three. Third process is osmoregulation. This is water absorption. Process number four that we don't really go into is secretion of toxins. So from the tissues around, we can get toxins being put into the tubule. And then that means there's more toxins in the urine going out. And then finally, and really it's kind of the fourth process that we talk about, we have more osmoregulation taking place in the collecting duct. And this is to do with ADH, that hormone from the brain. Okay, so let's zoom in then. This is ultrafiltration. Ultra because it's tiny. It cannot be seen even with a microscope. The scale that we're talking about, like with ultrastructure of cells, is tiny. And filtration because, like you can do in the biolab or in the chem lab, it's a bit like having filter paper and a filter funnel. When we put things through, we get a filtrate. So the filtrate is what we get through, and then there are things that can't get through. They are too big, maybe. Well, actually, let's draw these as a protein and a red blood cell and maybe a white blood cell. Okay, so things are too big to get through, they get stuck. Other things that are small, like salts, and things like we would have as well H2O, we'd have water in there, salts, glucose, things that are too small will go through. Okay, so let's try and follow through then. We have over here blood entering, and it's entering through the afferent arteriole, and it's exiting through the efferent. And what does that do? Well, because we've got a larger blood vessel entering in the form of the afferent arteriole versus the smaller efferent, we get an increased pressure pushing out. And that's going to help in ultrafiltration. So around the outside we can see Bowman's capsule, which is going to collect our filtrate. So this is what's coming out, the filtrate. Bowman's capsule collects it, and then the blood leaving has had its concentration of different things like glucose and salts and toxins and water. The concentration has changed. Also, we talked about lower oxygen. Okay, so let's zoom down here. We can see, although it's pretty hard, let's zoom in slightly more. We can see down here that we've got the wall of the capillary. This is the capillary wall, and it's got pores in it to allow things through. And the second wall, because it's the final barrier, we can call this, which is labelled there, the basement membrane. Okay, so around it you can see, so there are pores through which things can go. Now, around it you can see we've got these other cells. These are cells called podocytes. And they're kind of weird because they reach around and wrap around the capillary. But between them, you can see maybe just here, there are tiny gaps. So there are also pores between the kind of legs or the arms of the podocytes. So we could say they're filtration slits. So filtration slits. So we've got these two kind of pores, and that means that these things, which are big, cannot fit through. Plasma protein, red blood cell, blood platelet. So if we get blood in the urine, that's a bad sign. All of these things, glucose, amino acids, water, they are small enough they can fit through these pores. So that's up to the start of the recap at the ultrafiltration video.